All right. We talked about cytoskeleton. Now we're going to talk about ribosomes. So these are another group of non-membranous organelles. Okay. So that means they come in contact with the cytoplasm itself. And basically what a ribosome is just a globule of ribosomal RNA, rRNA. So basically if you take an RNA molecule, so here's our RNA molecule, remember it's only one strand, and you take it and you just globule it up into two main parts. That's basically what your ribosome looks like. I mean, seriously. You know, because it's so small, it looks like one contained piece, but it's just RNA that is folded on itself. Okay, so it's a globule RNA. So what does the ribosome do? What does it do? It is responsible. It is responsible for building. It builds the polypeptide the polypeptide of protein synthesis. Okay, so we talked briefly about genes, about what a gene is, and a gene is what codes for a protein. Okay, so now when DNA says, okay, we need to make a protein, what it does is it makes this thing called mRNA, messenger RNA. And messenger RNA, what it does is it goes to the ribosome. And so what the ribosome does is the ribosome reads this message, and basically it assembles the protein based on the message that mRNA is giving it. So that's what ribosome does. It assembles the protein. And remember, proteins are nothing more than long chains of amino acids. So what the ribosome does is it assembles that chain of amino acids. Okay, so that's what it is. It's a polypeptide. There are many amino acids in the chain of a protein. Okay, so the ribosomes come in two types. Okay, there are these things called free ribosomes. Okay, things that are called free ribosomes, and what they do is they just float in the cytoplasm. Okay, they just float in the cytoplasm. Okay, so they're going to build proteins in the cytoplasm. Now, one thing that I want you to take away from this, and I want you to remember this, cytoplasm is predominantly water. So the proteins that the free ribosomes build are totally compatible with water. Okay, so water-soluble proteins, or they can interact with water pretty well. And then we have these other ones that are called fixed ribosomes. Okay, we have free and we have their fixed. Fixed ribosomes, they're attached to this thing called the endoplasmic reticulum. Or the ER. And when you have ribosomes attached to the ER, it is called the rough ER because it looks like sandpaper. So if we look at a cell, so we're going to draw another cell here. So we look at a cell, okay, and so we've got a nucleus right here. So there's the nucleus of the cell. Ribosomes, here's how big ribosomes actually are in the cell. That's it. They're really tiny. And actually, I think my little points are too big. Okay, they're really tiny structures. And those would be free. They're just floating around in the cytoplasm. Now, attached to the nucleus here, this looks like an egg. But I guess that's a good example for a cell. Attached to this nucleus here, you have this floppy membrane thing. And that's called the endoplasmic reticulum. And attached to this endoplasmic reticulum, you'll have ribosomes. And that's what a fixed ribosome is, if it's attached to the edge of the endoplasmic reticulum. Usually it's on the outside edge of the endoplasmic reticulum. Okay, so The endoplasmic reticulum is membrane bound. We're going to talk about it when we talk about the membrane bound organelles. But membrane bound organelles are separate from the cytoplasm. They are separate. And what the endoplasmic reticulum does is it has a non-water based center. You know, so if I'm going to build a protein inside, if I'm going to build a protein on a fixed ribosome, it's going to be put into the endoplasmic reticulum. Okay. Usually these types of proteins do not like to interact with water, and water will actually harm them. So that's why we have ribosomes on the endoplasmic reticulum. Okay. Now that's just a general rule. And, you know, anybody who watches this, you can probably come up with a counterexample saying, well, blah, 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 blah. Fine. That's okay. Do your specialty thing. But in general, free ribosomes are going to make more of our water soluble things that can interact with water. The free ones will. And the fixed ones, they're going to make the ones that really don't like to. And they're packaged for very specific reasons. Okay. So that is the ribosome. So if you want to see a detailed picture of the ribosome itself, it actually has a small portion. It actually has a small portion attached to it, and it has a large portion. It has a large portion itself. Okay, right. So that's it. And so what happens is usually the small portion can attach, and what you do is the mRNA will come in through like this. And then the ribosome will attach. The small portion will be on top of the mRNA. The other portion will be on the bottom. And you can read it. You'll read it. And the small portion actually has like 3 channels. It's got like 3 little gaps in it. And the amino acid will come in and it'll attach like that. So, and then we'll talk about that when we talk about DNA replication. But I just want you to understand what a ribosome is. What it's made of. It is made of globules of RNA and, let's just say, other proteins, just to be specific, to be complete. Okay, and where are they found? You find them in 2 places. The free ones just float around the cytoplasm. Fixed ones are attached to the endoplasmic reticulum. Okay, and what do? Protein synthesis. They assemble the protein of protein synthesis. Done and done. Okay, so there's one more non-membranous that I want to bring to your attention. Okay, and it's called a proteasome. A proteasome. Okay, and so basically these are just enzyme-filled globules, and they are enzyme-filled globs. Okay, and what their job is to recycle proteins. Okay. Okay, the enzyme inside of a proteasome is called a protease. Okay, the enzyme contained in are called proteases. Okay, and what they will do is they will target damaged proteins and they'll break apart the proteins. And basically what this will do is it will recycle the amino acids, so the amino acids can be used later. And one of the things that the body doesn't do is it doesn't waste anything if it doesn't have to. So you'll degrade proteins all the time and just recycle them for later. Okay, and with that we are finished with the non-membranous organelles. Okay, done and done.